Okay, the lab we're going over today is diffusion and osmosis, and this is going to be another follow-along lab very similar to the one that we did over pH. So your job is going to be to have this lab out and available, and then you're going to follow along and you're going to jot down the data points that we go over as we go through the lab. As always, with any lab you get, because you're probably going to have to take science courses in the future, print your lab out and read through it. Right? Before you even come into class, read through that lab. In fact, a lot of instructors incentivize you doing that by having a pre-lab quiz. Now, we don't have a pre-lab quiz, we have a post-lab quiz, meaning after you do the lab, there's going to be a quiz over the material covered in the lab. But a good idea before you get going with any lab is to read through. So, we look at the materials required. We would have it on a cart or on a tray. We would kind of organize ourselves, look at the different things, maybe place the items for one experiment here, there, another place, just so everything's in good working order. And then you have your introduction, which gives you a conceptual introduction to what we're going to be talking about, which is essentially diffusion and osmosis. So we've gone over that in class, and I'm not going to cover it again here, but it gives you a good background, a nice concise description of what it is we're actually going to be looking at during the lab today. Now, in activity 1, we're observing the diffusion of a dye through agar gel. Now, as you're going to see in the video, agar gel is this porous polysaccharide jelly that allows for diffusion to occur because it has lots of nooks and crannies. One of the things that we're interested in are the different factors that influence the rate of diffusion. So we talked about those factors in the presentation that you guys had, the lecture presentation, but things like the temperature, particle size, the steepness of the concentration gradient all play a role. With this particular activity, I would think particle size. Have particle size in the back of your head. Now, this is the data collection point, and what I did in the video is I collected the data at point 0 and at point 60. So you're not going to have to jot down data points every 10 minutes. Just jot down the beginning and the end data point, and that's enough to answer the lab questions that you have. Now, in activity 2, we're observing diffusion and osmosis through a non-living membrane. The membrane we're using is what's called dialysis tubing. And dialysis tubing is a selectively permeable membrane, so it's really good to kind of mimic the characteristic of cell membranes because it allows some stuff across but not others, and a lot of times that's dependent on size. You have what are called molecular weight cutoffs, and we don't know what the molecular weight cutoff is for our dialysis tubing, meaning we don't know how big a particle can be and still fit through the little holes in the tubing. So we're going to evaluate that. We're going to evaluate two things. So you can read through that, and then when you're watching the lab video, you'll have an idea. We're essentially going to make an artificial cell out of dialysis tubing, and we're going to do two things. I'm going to get the weight of that artificial cell given the different parameters, so make sure to look at what's inside the cell, right, and what's the solution that the cell is soaking in, because we're trying to define tonicity characteristics, and if you understand tonicity, you'll kind of understand what the expected results of this particular lab activity are going to be. But I'm going to get the weight of my artificial cell initially. Then I'm going to get the weight of my artificial cell after it soaks for 1 hour. I'm going to get my delta change in weight, and remember we're taking weight in grams, so you're going to see that, and we're going to see the change. I'm not going to give you the change. I'm just going to collect this data point and this data point. Your job is to determine the change. And then I'm going to use silver nitrate to evaluate for the presence of sodium chloride in the solution that the cell was soaking in. So the cell begins by soaking in a solution of distilled water. Distilled water is 100% water. And then what we're putting inside the cell is a 10% sodium chloride solution. So we want to look at the movement of water. We want to evaluate osmosis. We also want to evaluate the permeability characteristics of the membrane for solutes like sodium chloride. The way we do that is using a test indicator. So our test indicator is silver nitrate. Now what I mean by that, and I'm going to go here to just a different presentation real quick, is I want to know whether the membrane that we were using, that dialysis tubing, whether it's permeable to sodium chloride or not. Presumably, sodium chloride, if it is permeable, is going to diffuse from areas of high concentration to areas of low concentration. So if that membrane is permeable, we're probably going to find sodium chloride in the solution. It was distilled water, but did any sodium chloride get into that distilled water through the dialysis tubing? That's the question we're really asking. So we use silver nitrate as an indicator. We're using silver nitrate to test for the presence of sodium chloride in what was distilled water. So the reason that silver nitrate is useful is when you dissolve silver nitrate in water as its parent compound, the silver dissociates from this nitrate and you have two independently floating particles and the solution is completely clear. Same thing with sodium chloride when it dissolves in water. The sodium dissociates from the chloride and you have a sodium ion floating independently from that chloride ion and the solution will appear completely clear. Now, when these two react, when the silver, which is denoted with the chemical symbol A lowercase g, reacts with the chloride, if it's present, meaning that if sodium chloride is present, silver is going to react with chloride, and silver chloride is not soluble. So it's going to form what's called a white precipitate. It's going to When silver and chloride react, they bind to one another and they form a non-soluble precipitate that's going to drop out of the solution as a solid. So it's going to form a solid and you're actually going to see it physically come out of the solution. You're going to see the solution change colors to kind of a silver color and you're going to see that precipitate, that non-soluble stuff, kind of drop out of the solution. And then the sodium nitrate is soluble, so you're not going to see that. And what this indicates, if you do get that precipitate reaction, is that sodium chloride was present in your solution. So it tells you about the presence of sodium chloride, which tells you about the permeability characteristics of our artificial membrane. So when you look at this reaction, this little GIF, before you drop the silver nitrate in, mark the color. So before, completely clear, drop the silver nitrate in, that silver reacts with that chloride, forming silver chloride, right? That silver chloride is not soluble in water, so it's going to drop out as this white precipitate, and it's a very obvious reaction that takes place if you have sodium chloride present in this solution. So that's it until we get into the lab. Yay! Okay, so what I'm going to use to introduce the methylene blue is a little micropipetter. That's what this plastic tool right here is called. It can be very hard with the dropper sometimes to get a consistent dot, and I just want a little dot of methylene blue on here to measure initially. So there's our dot of methylene blue. Notice that I have a few errors over here in practice because this can be a bit tricky to do. We're trying to standardize our dots as much as we possibly can. So I may even take this over here and get a little bit of a flow going, and then I'm going to try and standardize the smallest drop of potassium permanganate I can possibly get. Now those two dots are relatively equal, so what I'm going to do is I'm going to measure them right off the bat. Now remember, we're measuring in millimeters, so pay attention to where it says inches, centimeters, and each one of those little lines in between the centimeter marks are millimeters. So the initial size of this dot, to my best estimation, of the methylene blue is 5 millimeters, and of the potassium permanganate is around 4 millimeters. So the potassium permanganate dot is a little bit smaller than the methylene blue. What we're going to do now is we're going to wait 45 minutes. We're not going to actually watch this for 45 minutes, but we're going to come back and we're going to look at it through the magic of video. Okay, and through the magic of video, then we have the final size of our dots. So now the methylene blue and the potassium permanganate have been sitting in that agar gel for 45 minutes. So we're going to measure the size of the methylene blue dot. It's around 6 millimeters. Now notice that the potassium permanganate dot looks much more transparent, right? And that's because the particles aren't all concentrated in one area. They've diffused out. So they diffused vertically into the agar jelly. They've diffused horizontally away from the initial point. So when we measure the potassium permanganate again, 14 millimeters. The diameter of that dot is around 14 centimeters if we go out to the very lightest of the halo. So there's your initial, there's your final. That's enough to answer the lab questions that you have over methylene blue and potassium permanganate. Keep diffusion and the factors that influence diffusion in mind. Thank you so much. Now in the second part of today's lab, we're going to be looking at the permeability characteristics of membranes by using an artificial membrane to kind of evaluate how we can figure this stuff out. So the artificial membrane we're going to be using today to create our cell is called dialysis tubing. And dialysis tubing, which we're pulling out right here so you can kind of see right there, is interesting because it's porous, whether you can see those pores or not. And depending on the size of the pore will dictate the size of the particle or the molecule that can fit through. It's called the molecular weight cutoff. So the lower the molecular weight cutoff, the smaller the pores. The higher the molecular weight cutoff, the larger the pores. Larger pores allow larger solutes through. Now, dialysis tubing, just like a cell membrane, is a selectively permeable membrane, meaning that it allows certain stuff across but not other stuff. So we're going to evaluate the permeability characteristics of this membrane, and in doing that, we're going to look at the processes of diffusion and osmosis. So the first step in today's lab is creating a little artificial cell. Now in order to create an artificial cell, you kind of have to open up your dialysis tubing. And you do that by coming to the end, kind of like it's a grocery bag, and rubbing it in between your fingers, and you'll see the dialysis tubing actually pop open. Now these clamps, which most certainly don't go in the garbage if you're doing this lab in person, I'm going to put one of these bag clamps at the end of the bag on the opposite end from which I just opened it. Now, the instructions in the lab say to take a certain volume of water. It doesn't really matter as long as you get the initial and the final weight. So we're going to just say 10 milliliters of water that we're going to be putting in this. Now, remember that what we are putting in the actual bag itself is the 10% sodium chloride. So we are putting the 10% sodium chloride solution into the bag. 10% sodium chloride solution going into the bag. So I'm going to take this off, and I'm not going to use my dropper here. I'm actually going to use my micro pipetter. So I'm going to take the top off of that. Now this is a 3 milliliter micro pipetter. Every one of these little lines, horizontal lines across the side, indicates a milliliter, so I'm just going to use multiple of these so I can standardize my bag a little bit more carefully. So there's two, whoa, it'd be nice if I had somebody to help me hold this bag open. Good thing you guys will when you have your groups. Okay, so I'm going to start getting solution in there so it's a bit easier to open my bag. Mistakes are part of the game. Whoa. Oh. Oh. Whoa. 2. There we go. 3. 4. Oh, loaded. That is not easy. 4. And then, because I'm doing 2 milliliters of pop with each one of these pipettes, we're going to go 5. So we put approximately 10 milliliters of a 10% sodium chloride solution into our artificial cell. Now what we're going to do is we're going to clamp off the other end of the artificial cell. Now before doing that, I didn't do that very well, but that's good. It's a learning moment for you. I'm going to try and remove as much air out of there as I possibly can. So I'm going to go back, and I made a little oopsie, and I'm going to try and remove as much air out of that artificial cell as I possibly can. There we go, much better. Not much air in there, we're good. Now, I'm going to weigh this artificial cell initially, and before I weigh it, just so I don't have residual water influencing the mass or the weight of the cell, I'm going to kind of pat it off with these paper towels. Then I'm going to turn on my scale. Going to wait for it to zero out here. Then I'm going to pop on my weigh boat. Now remember, after you pop on your weigh boat, because you don't want to be accounting for the mass of the weigh boat, you can zero that out. Zeroed it out. Now I'm going to weigh my cell initially. So my artificial cell initially weighs 21.25 grams. So my artificial cell initially weighs 21.25 grams. Make sure to mark that down. Now, through the magic of movies. Okay, now what we're going to do is we're going to place our artificial cell. So we determined its initial weight. We're going to place our artificial cell into a solution of distilled water. So we have distilled water, or 100% water, and within the cell we have a 10% sodium chloride solution. So we got our initial weight. Make sure to write that in your lab values because you're going to be submitting this lab report to me. And now I'm going to soak this cell right in distilled water, and you can take even a little more because the volume of the distilled water doesn't matter. We're going to soak this cell in distilled water for 45 minutes. Now, after 45 minutes, through the magic of movies, we're now going to weigh this cell again. So we're going to take this cell out. It's been soaking in that solution for 45 minutes, and we're going to weigh this cell again. Remember, we always want to zero out when we have our weigh boat on there. Pat our cell dry. The final value, or the final weight of this cell after it's been soaking in there, is 23.05 grams. So we have 23.05 grams. So those are your data points that you need to answer the first set of lab questions. Now, in the second set of lab questions, we're going to actually look at our solution. So we weighed our artificial cell before and after. Remember, inside the cell there was a 10% sodium chloride solution, and we soaked that artificial cell with that dialysis tubing membrane, that selectively permeable membrane, in distilled water for 45 minutes. So we used our timer. We actually set our timer. You can just enter 4500 and press start on it, and we set our timer and we waited 45 minutes. Then we took that cell out and we weighed it again. Now, based on the initial weight of the cell and the post-solution soak weight of the cell, you can start answering questions in your lab. The final set of questions in your labs relates to this experiment that we're performing now. So we want to determine whether the membrane of that particular, that dialysis tubing membrane is permeable to sodium chloride. Now remember, this initial solution that we soaked the cell in was distilled water, meaning no sodium chloride there initially. Now we want to determine whether sodium chloride is present or absent. And the way we do that is we use another chemical indicator that reacts with sodium chloride called silver nitrate. And what silver nitrate does is there's a double displacement reaction in which silver nitrate reacts with sodium chloride to form a solid precipitate called silver chloride. And if silver chloride is present in the solution, it will actually go from being a aqueous form of silver. It'll bind to that chloride, and that silver chloride precipitate will fall out, and we'll see it turn kind of a silvery color. We'll see the solution turn color. Right? If that solution does turn color and that solid precipitate falls out, it means that sodium chloride was present. And what does that mean about the permeability characteristics of that dialysis tubing? So what I'm going to do is I'm going to take a small volume using my micropipetter of the actual solution that we soaked the cell in, which was originally just distilled water. And I'm going to put it into this test tube over here. And to make it easy to see, I'm going to add 6 milliliters to this test tube, even though that deviates a bit from the lab protocols. These pipettes are kind of weird, so I always just use that 2 milliliter mark right there. And then I'm going to take this silver nitrate, and the volume doesn't matter because silver nitrate, if there is no chloride ion present, is just going to go into solution and you're not going to see it. But if chloride is present, it's going to react with that chloride. So I'm going to boom, boom. Now if you look at this test tube, you'll see that that silver is reacting with the chloride ion to produce a solid precipitate that falls out of the solution, and that's why you see the solution becoming cloudy. That means that chloride ions were present. So we started with distilled water, but now in that solution that we soaked the cell in, we have chloride ions present. So what does that tell you about the permeability characteristics of the membrane? That's for you to answer. That's it. Have a good one, guys.